Good afternoon to all. My name is Marco Pelo and my presentation is about the subject of my PhD project, the diversity and evolutionary history of Ateuchus, a clade of New World dung beetles. Dung beetles are well known among us beetle experts for their coprophagous habits. Indeed, many of their synapomorphies are adaptations related to these feeding habits, including the soft mouthparts as illustrated here by these mandibles. There are more than 7,000 species worldwide, and the fossil record dates back to the Eocene. The systematics of New World dung beetles has been extensively studied over the past decades, but three genera, for their sheer number of species, are still great taxonomic challenges in the group. My project deals with one of them, Ateuchus. Ateuchus is a widespread taxon in the New World, ranging from the US to Argentina. Each species vary in length from about 4 millimeters to about 16 millimeters, and although most of them are shining black, some show vivid metallic coloration. Unlike many well-known groups of dung beetles, males of Ateuchus don't bear long cephalic or pronotal horns, have at most a low cephalic carina in both sexes. Sexual dimorphism in Ateuchus is more common in the pygidium, which in many species has carinae or spines in males only. The function of such structures is unknown. Indeed, very little is known about the biology of Ateuchus. The nesting behavior of only 3 species have been described, and the immatures of only 4 species are known. There are some species that are known to be myrmecophilous, some of them perhaps even obligatory. Others are incline of vertebrates, particularly rodents and the gopher tortoise in Florida. When we started this project in 2018, 103 available names were included in Ateuchus, 96 of them considered valid. However, the last and only general revision made for the genus dates from 1868 by Edgar von Harald, the great scarab expert from the 19th century, who then included only 28 species in Ateuchus. Since then, most species have been described in isolated papers. Exceptions are the faunas from North America and Costa Rica, which have been revised by my colleague Bert Coleman. For the rest of the continent, almost nothing is resolved. We don't know how many species there are, how to identify them, where they occur, or how they are phylogenetic related to one another. Furthermore, the close relationship between Ateuchus and the Scatimina, as proposed in 2007, has more recently been called into question. In turn, it has also been suggested that 2 small South American genera, Aphendium and Deltorhinum, are likely highly modified lineages nested within Ateuchus. Therefore, if this is correct, Ateuchus would be paraphyletic. But is this correct? Is this suggestion correct? To explore all these questions, my project aims to revise the complete systematics of Ateuchus based on evolutionary principles. And these include the investigation of its alpha systematics, phylogeny, and biogeography. But for this presentation, I will concentrate on the first part only. To conduct this part of the project, I have been relying on museum specimens. I have so far studied 65 collections from the Americas and Europe, which together totalize more than 44,000 examined and identified specimens, including the types of most of the names. For the delimitation of species taxa, I have followed the biological species concept. Therefore, when I say that a given group of population constitutes a given group of populations constitutes a species, what I mean is that my hypothesis is that that group is a genetic cluster separated from other such clusters by biological isolating mechanisms. If this is later shown to be incorrect, then my hypothesis will be falsified. For the limitations methods themselves, I have followed the traditional guidelines of those who adopt the biological species concept. Evidence supporting my hypothesis are from morphology and distribution. More specifically, mostly from features that are traditionally regarded as evolutionary classic among dung beetles, such as the male genitalia and the secondary sexual characteristics. For the phylogenetic analysis, however, we will integrate these plastic features to the more conservative ones from the mouthparts and wing venation. So as a result, including some transfers from and to Ateuchus, we have concluded that 122 already available species group names belong to the genus, 90 of them denoting valid species. Additionally, 54 new species have been discovered. In total, therefore, Ateuchus includes 144 known species. My museum studies have also resulted in more than 3,500 geographical records from more than 2,000 localities. And the species are, of course, not even distributed across the continent. As you can clearly see here, the species richness is highly concentrated in the Amazon region and, to a lesser degree, in the Cerrado and in part of the Atlantic forests of Brazil. And you can also see here that the bulk of the unknown diversity is concentrated in inland regions of South America, particularly in Brazil and Peru. And other general facts can also be stated. Here you have new and already described species according to the number of known specimens and year of first collection. You can clearly note that the more abundant species, as of course expected, were the first to be collected and described, although there are some remarkable exceptions. For instance, the third most abundant species, which was first collected in 1829, is a new species. Another interesting fact is that new species continue to be discovered almost every year. 14 species have been first collected in the past 20 years alone, suggesting that more are yet to be discovered. Therefore, the monograph is probably far from being complete. On the other hand, you can see here that 5 species have not been collected for more than 60 years. 2 of them last seen in the 19th century. One that deserves special attention is Ateuchus asperatus. All of the 20th century specimens were collected in a Restinga area in the neighborhood of Galeão in Rio de Janeiro. Nevertheless, this area has been greatly altered for the construction of the Rio International Airport since the 1970s. So is it possible that the species is now extinct? I think it is a real possibility, at least for at a local scale. A remarkable fact observed by us among the species of Ateuchus is the existence of morphological clines along the range of several widespread species. These clines concern characters like punctation, shape of the pygidium ornamentation, shape of the endophytes, and coloration. Interesting example is the species pair Connexus and Irinus. In the area where they overlap in Western Amazonia, they are very similar to each other. However, eastwards, they are limited, where they are limited either to the north or to the south of the Amazon River, they gradually diverge from each other in several characters. In another species, Ateuchus semicucleus, the populations are shining black in the Brazilian savanna, but coppery in marginal areas of northeastern Amazonia. However, there is a gap between these populations, likely a result of lack of collections. How is the call of these unknown populations like? We can only answer this question with more field work. And finally, there is an example that deserves to be discussed in more detail. Ateuchus steroides was divided into 3 taxa based on the combination of the states of 3 characters. However, we have observed that the characters used to delimit them are not only clinal, but their clines are not synchronized. Some of these clines are very wide, such as the one concerning the pronotal punctation, having a rather smooth intergradation, while others, particularly in Florida, are shorter and more abrupt. In addition to these 3 characters, we found a 4th cline, this related to the male genitalia. For this reason, we consider that these populations constitute a single widespread polymorphic species instead of 3 separate taxa. But what is the origin of these clines? We have tried to formulate some initial hypotheses to be tested in future projects. As you can see here, Ateuchus steroides is present mainly in temperate forests. It is known that in the last glacial maximum, the North American temperate forests were reduced to small fragments in the southern US. Our hypothesis is that either declines may be secondary, in which case the populations initially evolved in isolation and from one another in this refugia and later regained contact, or that these clines may be primary, a result of the expansion from a single refugium after the last glacial maximum, with subsequent local adaptations and divergence by distance. And of course, it can be a combination of both. I hope to answer this question in future projects. And there are many other patterns of variation worthy of further investigation among the Ateuchus using molecular, ecological, and morphometrical approaches. These patterns include interspecific ones, such as the ones exemplified here, as well as interspecific ones that, when better understood, can shed some light on the nature of the speciation process among the dung beetles. So to conclude my thesis, we will include sections summarizing the entire knowledge about the 144 species of Ateuchus, including information about the types, breed descriptions, of course, information about ecology, geographical distribution maps. And the next phases are now exploring the phylogeny image biogeography of these 144 species. Particularly interesting is the participation of Ateuchus in the Great American Biotic Interchange. I want to know how many species migrated northwards, when they did so, and whether it was before or after the uplift of the Isthmus of Panama, and this assuming that Ateuchus originated in South America, as most dung beetle specialists assume they did. So I'd like to thank all those institutions that provide funding for this project to happen, to my supervisors for their support and trust, to the Collectory Society for the invitation, and in special to all the curators, collection managers, and collectors, the people who are making this work possible. Thank you very much for your attention.